discuss about which topic, ma? We discuss about major histocompatibility complex. What is meant by major histocompatibility complex? Major histocompatibility complex is nothing but it is or set of organization. The set of organization is takes place in between the nucleated cells. What is meant by nucleated cells, ma'am? The complex molecules itself, they are having the nucleolar region in the cells. Here is major histocompatibility complex is present in the surface proteins which are located on the cell membrane of nucleated cells, and it plays a major work or major important role, more important role in the identification of self and non-self antigens, which why, because of in our body our organization system defends against the foreign antigens. So this major histocompatibility complex helpful for the detection of foreign antigens, which means non-self antibodies, and it is also responsible for the intracellular recognition of the antigens, and these are also responsible for the antigen presentation, which means for example, if we observe some of the microorganisms are enter into our body, at that time these cells are recognizes that antibodies, and they present these cells to the T helper cells, which means immunological cells which are present in our immune system. Okay, here histo refers for cells or tissues, the compatibility with which is refer for them living together. Okay, which means the organization setup which is present in the surface proteins of the cell membrane in the nucleated cells are classically called as major histocompatibility. Okay, here MHC is always recognizes only T lymphocytes. Okay, T lymphocytes lo మాత్రమే ఏం జరుగుతుంది ఇక్కడ recognition will be takes place. Here there is two types of immunity based on their MHC presence, which means T helper cell recognized major histocompatibility, and another one is T cytotoxic cells recognized by MHC1 molecules. Okay, here coming to the history of this major histocompatibility, uh, Peter Greer in 1930 found that there are four groups of major histocompatibility molecules. He used it the blood samples of mice and identified the blood group antigen which is designated by 1 to 4 groups of MHC classes. Here the classification of MHC takes place by Peter Goyer. Okay, in 1930, he recognizes such type of molecules which are present on the surface of the cells. In that, especially he observed in the mice blood samples, there are four types of major histocompatibility complexes are present. Okay, and another discovery also is there. Gregor Snail and Jean Desert and Berry. These three people are getting received Nobel Prize in 1980 for their contribution in the discovery of MHC. In the discovery of MHC molecule. Here there are MHC is categorized into four types. They are namely MHC1, MHC2, MHC3, MHC4. Here, first class of major histocompatibility class complex, and second one class 2 major histocompatibility complex molecules, and third one is class 3 major histocompatibility molecules. Here if we observe that the major histocompatibility for molecules. Okay. Here if discussed about the detailed classification of this MHC molecule. Class 1 molecule. First one is class 1 molecule, and this class 1 molecule is a group of histocompatibility, major histocompatibility antigens, and this is consisting of 45 kilodaltons molecular weight. Okay, they are present in the surface of nucleated cells, and they are responsible for involving in recognition of foreign antibody. They are also present on the surface of all nucleated cells except nerves, tissues, and platelets, and it presents antigens into the T helper cells. Here, the organization will be takes place in between the T helper cells and T cytotoxic cells. Okay, and it brings about the cell mediated immune responses in the MHC1 complex. Okay, at the mouth in the girls, are you clear? Hello, hello, girls.
Madam, అర్థమవుతుంది.
At the mouth. Okay, good. Then coming to major histocompatibility complex 2. For example, if you observe this, I share one photo. Yeah. Here, for example, if you observe this MHC class 1 molecules, sorry, if you observe this diagram, here these MHC class 1 proteins, they are consisting of a peptide bonding groups. One is alpha and one is MHC molecules are consisting of two polypeptide chains. One is alpha, another one is beta chains. Okay. And alpha chains are which are non-covalently attached with beta 2 microglobulins. Okay. These alpha chains are consisting of a transmembrane proteins which are encoded by A, B, C genes in the group of HLA. For example, if we observe in this diagram, three alpha proteins are present and one beta proteins are present in this diagram, and three alpha globulins are which are non-covalently attaches with beta anti. Okay, beta microglobulins. Okay, and when antigen interconnected and produce a proteasome structures, these peptides are produced in that type of peptidization, further loading of group in MHC1 molecules from the endoplasmic reticulum. Here, these peptides are further loaded on the groove of MHC1 molecules from the endoplasmic reticulum. Okay. Here, second one, MHC class 2. Here the MHC class 2 molecules, a major histocompatibility molecules are present in the surface of antigen presenting cells. Okay, these antigen presenting cells which engulf the foreign antibodies, which means they are not representing to the T cells. They directly invaginate or engulf them for foreign antibody, foreign antigen, and it binds with the endocytic degradation of antigen, which means the cell itself engulfs inside the micro the foreign antigen itself engulfed by the this MHC molecules, and it shows the endocytic degradation. Endo means what? Cytic means cell. Cell inside the cell, the degradation of these foreign antigens are takes place. Okay, and it binds with CD4 adenosine molecules of TH cells, and it is also consisting of two polypeptide chains, namely one alpha chain and beta chain. Okay, these antigens are processed in the inside the endosome, and the peptides are further loaded with MHC groove molecule. For example, if we observe again this diagram, here there are two types of peptide grooves are there. One is alpha, another one is beta. They are directly attaches with the cytoplasmic cells. Okay. Okay, ma. కనబడుతుందా? Hello. Hello. Girls. And MHC3 molecule and MHC4 molecule. The molecules include the complements like C2 and C4 and BF factors, means factor B are present in the MHC3 molecules. And the class 4 molecules are consisting of T cells. They are present these molecules present the T cells of leukemia as well as immature thymocytes. Here, MHC4 molecules are present in the T cells of leukemia, and as well as it also uh, as well as an immature thymocytes. These are present in the T cells of leukemia and as well as in the immature thymocytes. Okay. Then next coming to this next slide. What is meant by humoral leukemia? Okay. Okay. Are you clear, girls? ఇక్కడ వరకు here the HLA means human leukocyte antigen. HLA means human leukocyte antigen. This human leukocyte antigen which is present in the human beings, and it is present in the set of surface on the nucleated cells. Okay, they are the responsible for the graft rejection, adaptive immunity, defensive mechanism against the foreign antigen, and sometimes it is also exposed to, on cancer cell destruction and certain autoimmune diseases and such complements, which means HLA is highly responsible. The more important plays an important role in the graft rejection, adaptive immunity, defend against infection, and sometimes it is exposed on cancer cell destruction and certain autoimmune diseases. which means these cells are helpful for the graft rejection. What is meant by graft rejection? Okay. Do you know about transplantation process? What is meant by transplantation process? Transplantation process is nothing but here the grafting is takes place from one organism to another organism. Grafting is nothing but a patch of tissues, which are transferred from the one organ to another organ. Another organ or tissues, which are transformation of tissues or organs from an individual to other, are the cell are known as transplantation process. The tissues or organs which are useful for this transplantation is called as grafts. Okay. Here the graft rejection means if the organisms are not consisting of similar type of blood grouping and similar type of cells, such type of grafts are rejected. For example, if you take some of the persons are itself committed suicides. At that times, they commit suicide by burning their bodies with petrol or kerosene. At that time, the cell tissues are damaged on the surface area. The doctor itself suggests them to go for plastic surgery. In that time, they take the graft from the same similar organism, similar person from the thigh tissues. Okay. At that time, thigh tissues are used as graft. Sometimes the graft rejection is takes place by the microbial action, or sometimes it may be affect the foreign antigens entry in the our body. And second one is defense against the infection of some cancer cells, which means it also responsible for the in the destruction of cancer cells in the body, and it also avoids the certain type of autoimmune diseases in the body by creating MHC complex. This MHC is general term which is referring in the cell surface antigens of all the vertebrates. Okay, are you clear? ఇక్కడ వరకు అర్థమైందా? Are you clear or not, better?